Hello friends, welcome back to the lecture on insulin. So today we will be talking about insulin signaling as relevant to diabetes. My name is Dr. Santosh Abraham. I'm a specialist registrar in endocrinology and diabetes at Scarborough General Hospital. Insulin receptor, it consists of two insulin binding units. So one is an alpha subunit and the other is two of them are alpha subunits and two of them are beta subunits. So it actually consists of four subunits. So the insulin receptor has two insulin binding alpha subunits and there are two catalytically active beta subunits. So in total there are four subunits. The alpha subunits bind to the insulin. The beta subunits are catalytically active. The alpha and beta subunits are derived from a single chain precursor. Once the subunits are formed, they are linked by disulfides into an alpha to beta 2 complex. When insulin binds to these extracellular alpha receptors, it activates the intracellular tyrosine kinase, because the insulin receptor is basically a tyrosine kinase receptor. The intracellular tyrosine kinase domains of the beta subunits are activated and it phosphorylates intracellular partners on specific tyrosine residues. After this, multiple classes of adapter proteins including IRS, SRC, GRB and others will associate with the receptor and propagate the downstream signaling. Once insulin binds to the alpha subunits, it activates tyrosine kinase of the beta subunit, which will phosphorylate some other molecules which has specific tyrosine kinase residues and which will result in IRS, SRC and GRP associating with the receptor and propagating the downstream signal. This leads to modulation of cellular metabolism, stimulation of mitogenesis and receptor internalization. What are the downstream events? IRS, PI3K and AKT are the three critical nodes of insulin signaling. IRS allows for interaction with phosphorylated tyrosine residues on the insulin receptor. It is closely related, and also the closely related IGF-1. It allows to dock the downstream effectors such as the PI3K. In short, IRS allows for the interaction between the phosphorylated tyrosine residues and the insulin on the insulin receptor and the IGF-1 receptor. IRS thus acts as a middleman between the insulin receptor and the IGF-1 receptor, and it helps to dock the PI3K too. PI3K will generate PIP3. It will activate several PIP3 dependent serine trione kinases such as PDPK1 and PDPK2, which will in turn activate AKT isoforms and other molecules like atypical pKases or insulin stimulated serine kinase, etc. But the activation of AKT isoform is the most important. Once the AKT is activated, it phosphorylates proteins that regulate lipid synthesis, glycogen synthesis, protein synthesis and apoptosis. If this AKT is disrupted, it will result in growth retardation. There are two isoforms of AKT. So if the AKT1 is disrupted, it results in growth retardation, and if AKT2 is disrupted, it results in insulin resistance and diabetes. So remember, it is AKT2 which is associated with insulin resistance and diabetes. This is a diagram of the insulin receptor, the insulin signaling. Actually, when insulin binds with the insulin receptor, it will activate the IRS and which will activate the PI3K. The IRS in fact acts as a middleman between the IGF-1 and the receptor, and it will activate the PI3K, and it will again lead on to the activation of AKT. So AKT, this is a crucial part of the insulin signaling actually, or what insulin does to the biochemically to the other tissues. Like AKT will in fact, activation of AKT will increase the glycogen synthesis, as you show via this GSK3 pathway, and also mTOR will be activated, which will result in protein synthesis. At the same time, there is a AKT mediated translocation of the endocytic GLUT4 vesicle to the cell surface. And what happens after that, when the GLUT4 is translocated from the cytoplasm to the cell surface, it permits glucose to enter in. And mTOR via mTOR can also act on the GLUT4 translocation, and mTOR via AMPK can influence the glucose transport through GLUT1 also. So it will the mTOR via AMPK will influence the GLUT1 expression on the recruitment of GLUT1 on the cell membrane. So the net effects are protein synthesis via mTOR, also protein synthesis via PAKT through the GSK, also glycogen synthesis through the GSK, and inhibition of gluconeogenesis by FOCAT transcription factors, the FOXO genes. Downregulating them will lead to inhibition of gluconeogenesis. Let's now talk about tissue-specific actions of insulin. The mechanisms behind glucose uptake in muscle and fat by insulin are now discussed. So insulin in the skeletal muscle and adipose tissue stimulates glucose uptake by translocation of GLUT4 from the intracellular pool to the surface of the cell, which I have already shown you in the previous slides. There are two proteins called TBC1D1 and Rab GAP. This usually inhibit the GLUT4 translocation. But when the Rab GAP is phosphorylated, this inhibition is relieved, leading to increased glucose uptake. So you can infer from this that when there is a insulin binding to the insulin receptor on the skeletal muscle and the adipose tissue, this Rab GAP is phosphorylated, and the inhibition on the GLUT4 translocation is taken away. The number of glucose transporters in muscle of insulin resistant persons are not changed, but the ability of the insulin to affect GLUT4 translocation is disrupted in insulin resistance. So it is the insulin's issue to affect GLUT4 translocation rather than the GLUT4 transporters numbers that will result in insulin resistance. However, insulin receptors can associate with CAP, which will activate TC10. The activation of TC10 can lead to GLUT4 vesicle translocation in the absence of IRS PI3 AKT activation. This is another important pathway, how insulin can cause GLUT4 translocation in the absence of IRS P13 AKT pathway. Through CAP and TC10, it can also cause GLUT4 translocation to a lesser extent. It has also profound effects on protein homeostasis via AKT-mTOR to promote protein synthesis and cellular growth. At the same time, it suppresses FOXO transcription factor activation. So it's a master regulator of protein degradation pathways. So by activating mTOR and by suppressing FOXO, it will promote protein synthesis and suppress protein degradation. What is the role on regulation of lipolysis and lipogenesis? Insulin signaling, it will result in increased lipid storage in adipose tissue by stimulating triglyceride synthesis as well as suppressing lipolytic triglyceride breakdown. It stimulates rapid and potent increases in adipose tissue glucose uptake. The glucose is converted into glycerol 3-phosphate, which is used to synthesize triglycerides from fatty acids. So this is one of the mechanisms by which it will promote lipogenesis in adipose tissue. Another mechanism is fatty acid transport into the adipose tissue is increased by insulin. It could be possibly due to the translocation of fatty acid transporters, translocation of the fatty acid transporters, or increase in lipoprotein lipase expression and activity. And glucose also activates carbohydrate response element binding proteins, the CREBs, to upregulate glycolytic and lipogenic gene in adipocytes. And overexpression of GLUT4 in adipose tissue protects against insulin resistance caused by diet-induced obesity, which is very important. So the regulation is first by rapid increase in the glucose uptake, which is converted into G3P. Second is by the fatty acid transport, which possibly is due to the increased translocation of fatty acid transporters or increase in lipoprotein lipase expression. And third is by glucose, which is independent of insulin, which activates the CREB to upregulate glycolytic and lipogenic genes in adipocytes. So that could be the reason for glycolysis and lipolysis, whereas the others, like the increased glucose uptake by the adipocytes as well as the fatty acid transport, could result in increased lipogenesis. So we all know about beta adrenergic signaling, which will increase cyclic AMP, and then it will induce protein kinase A activation, which will in turn lead to the activation of principal lipases in fat cells. One is hormone sensitive lipase and the other is triglyceride lipase. Insulin, by activating PDE3B, will reduce the cyclic AMP level and will thus inhibit the protein kinase A mediated activation of adipose tissue lipases and hormone sensitive lipases. Another mechanism by which insulin suppresses lipolysis are by increasing in the adipose tissue lipase mRNA and by increased expression of FSP27. And deletion of insulin receptors in adipocytes leads to a rapid increase in adipose tissue mass. Redistribution of fat into liver, this is very important. This is where what the insulin receptor resistance plays. And lipodystrophic diabetes. Hence, insulin regulation of lipolysis and lipogenesis in adipose tissue plays a critical role in storage of lipids, maintaining whole body insulin sensitivity and preventing metabolic disease. What about fat formation in the liver? SREBP transcription factors are the master regulators of lipogenic genes, including all of these like the FS synthase, lipoprotein lipase, acetyl coenzyme A carboxylase, and stearyl coenzyme A desaturase isoforms. These are all regulated by insulin at multiple levels in the liver. Insulin basically increases the expression of SREBP1C. So it will in turn affect the mTOR C1, which will again affect the IRS PI3K AKT pathway, resulting in enhanced lipogenesis. And now other important topic about the insulin regulation of hepatic carbohydrate or glucose metabolism. How is glucose produced by the liver? It is the production is regulated primarily by a balance between the action of insulin to suppress glucose production and glucagon to activate glucose production. So this two opposing factors actually dictate or actually determine how much glucose can be produced by the liver. Generally, insulin is dominant over glucagon. The ability of insulin to reduce glucose output from the liver is one of the important mechanisms for maintaining normal fasting blood glucose and normal glucose tolerance. By inhibiting glycogenolysis, insulin will also suppress glucose release from the liver, and this effect is dominant over glucagon. Also, insulin decreases endogenous glucose production by direct and indirect mechanisms. So in the direct mechanism, it inhibits glycogenolysis and gluconeogenesis by insulin receptor activation, which I've told you before. With the peripheral or indirect effect, there are two forms. One is that insulin will decrease the glucagon secretion to a great extent. It could be by influencing or affecting the alpha cells through various paracrine effects or through the systemic effects. When the glucagon secretion is decreased, it leads to the decreases the activation of glycogenolysis and gluconeogenesis. That is, less glucose will be produced by decreasing the glucagon secretion. The second indirect mechanism is to decrease the substrates for gluconeogenesis, such as alanine from protein catabolism and glycerol and free fatty acid levels, because it suppresses lipolysis in adipose tissue. So the effects of these will contribute to decreased substrate for gluconeogenesis. So endogenous glucose production is decreased by two mechanisms, direct and indirect. Direct consists of inhibition of glycogenolysis and gluconeogenesis through insulin receptor. Indirect is through decreased glucagon secretion and decreased substrate for gluconeogenesis. Hepatic insulin resistance plays an important role in hyperglycemia of type 2 diabetes mellitus. This impaired suppression of hepatic glucose output appears to be quantitatively similar to or even larger than the defect in stimulation of peripheral glucose disposal. This is an important point, because more than the peripheral glucose utilization, the impaired suppression is a culprit in the T2DM. Even there is a direct relationship between increased hepatic glucose output and fasting hyperglycemia. This suppression of hepatic glucose output by insulin is impaired at both low and high plasma insulin levels in type 2 diabetes patients, and even the hepatic glucose production is elevated early in the course of the disease. This shows a picture of the insulin and its actions on various peripheral cells, peripheral tissues. In the alpha cell, it will lead to glucagon suppression. In the beta cell, it will lead to increased survival proliferation. In the endothelium, it will lead to vasodilation through NO. In the cardiomyocyte, it will lead to increased glucose oxidation and hypertrophy and decreased autophagy. In the macrophage, it will lead to decreased apoptosis and increased cytokine release. Even in the brain, independent of the blood mechanisms, it has been shown to improve the satiety, mood stabilization and cholesterol synthesis. This slide shows the effect of insulin on tissues other than the muscle and adipose tissue, which I've already told you before. Thanks for watching this presentation.